the layers of the mustard blooms, the cereals are right behind it. If we can just catch an inch of rain today, the cereal's absolutely going to explode out of the canopy. I would call it 3 to 6 inches shorter than last year at the same time. Last year, I think we had a little bit of some warmer stretches in February that pushed it to elongate. I think in another 6 or 7 days, it's going to be really pretty to see how this looks. And as I was standing there, there's a few blooms still on the trees, but the bee pollination and the pollinator activity in this row was incredible. You'd walk into it, the bees would land on you, land on your shoulder, and then just leave to go back to the mustard. Absolutely incredible for help on the biology. Our second control window was the Max Organic Builder that we put in, a blend of oats and bell beans, peas, and vetch. Very popular for us here in the North State. The one thing about the Max Organic that stands out, it's a little bit lower of a canopy height than the SR mix, and that allows for a little bit better frost protection. It allows for just a little bit better biomass later on. Oats will make a crop with almost no moisture, so it's definitely a high use item for us. That picture was taken in 2020. The same row this year shows about the same height as last year, even though we've had less rain. Everything seems to be on course. I'm hoping for an inch of rain today. It'll make this stuff absolutely pop. If you can see the little peas and bell beans in there are just starting to really set tendrils and flower. I think by middle of next week, our peas are really going to have a lot of blooms, and the bell bean first flowers should be really starting to be visible. This will be really pretty, and I'm really excited to see what it does. You'll notice that they've done some cultural work in this orchard, and you can see where the tire tracks have straddled our little plot. Compared to the control row, you do not see the compaction or the tire prints like the other things that we were looking at. So very, very impressed by what this is starting to show for us for this year. Termination time, last year, we were running at about April 10th when we went through. Some guys were a few days earlier or a few days later. We talk about this because I'm kind of looking at this year's control windows. For some of our guys that have planted early, they've already mowed and terminated. In this particular orchard, with the weather that we're having, it looks like we'll be on the same course as last year, maybe 6 or 7 days earlier if the dry weather persists. Everybody's wanting to terminate these crops before the seed pods are set, so we don't get anything blown into the rows or seeds off target where we don't want them to be. With everything taking place, we're definitely going to collect samples for weight again, take soil tests to confirm organic matter, nutrient levels, and then we're also going to take photos to compare what the orchard environment will look like for harvest so that we can have a nice clean row. Nobody wants a bunch of extra baggage when it's time to pick up. Last year, our SR mix went 25 green tons per acre at 75% moisture, or about 2.5 tons of dry matter at 100% adjusted. We had tremendous pollinator activity. The residue dissolved over about 2.5 weeks with some irrigation. I think that the stems definitely helped water penetration. And when we went to replant this year, you could see the very small remnants of where the stems were from last year. Absolutely impressed with how this really kept the ground soft as well. When we went to go take soil samples in these rows of SR mix and Max Organic versus the control rows, the probe absolutely went into the ground better than the control rows. The control rows were packed extremely tight. The Max Organic Builder last year with the weather we had was just a little bit behind in yield. 14.4 green tons of residue, came out to 1.44 tons of dry matter an acre. Had medium pollinator activity because our little bell beans and peas didn't bloom quite as much as I was hoping for with that late rain or not much rain in February. Same thing, the product dissolved in about 2 weeks with irrigation, and the stems were definitely present to help water penetrate through the summer. As I talk about these cover crops at these different slideshows, some of you have seen them before and others are always asking me questions, about issues that they have with cover crops. One of the first issues most people worry about, especially in almonds, is frost. What do I do if we get cold temperatures as the bloom is on? Will it increase our cold zone? Will it make our cold zone be up in the canopy where some of the blooms are located? It's possible. Behind this slide, I have a couple of pictures of before and after in almonds and also in some walnuts showing planting timing and how it can help you adjust for your situation to what's best for you. The other question that a lot of people have is regarding pests. Everybody wants to know, if we plant a cover crop, are we going to have off-target pests? Are we going to have some other issue eating on the cover crop that could then go into the trees if we mow it? Or could we overwinter pest, where it doesn't allow winter sanitization to get rid of mummies and other problems that we want to try and get rid of for orangeworm control. The biggest thing that we see with the pest side of these cover crops is that you can drive through them. If we have guys that have to do bloom sprays or do a blight spray for walnuts, we've mostly driven right through the cover crop, and most of them are light enough. They don't truly build up with a lot of leaves in the orchard fan or orchard sprayer fans. They've been pretty solid, and you can mow them at any time you want to, whether it's all the way to the ground or 4 to 6 inches tall. And we'll show that picture as we get into that next slide. Water requirements are always a hot topic, especially this year in California. Most of the time we can get things started in October with 1 good irrigation, possibly 2 if things stay dry. As we get into the spring, this year we haven't had anybody irrigating cover crops except for 1 or 2 orchards on some incredibly sandy, dry soils. I think we've seen people want to try and build their profile more into the tree root zone of 6 inches to 2 feet. And that's where we see a lot of guys watering early, trying to keep their soil probes in their green lines on their moisture meters rather than helping cover crops. Most of the cover crops on the water requirements are kind of an afterthought, as our trees are mostly asleep in January. And well, once they start to break dormancy in February, then it's off to the races depending on Mother Nature. Crop competition is always an interesting factor, especially when trees are young. Will these cover crops get taller than the tree rows that they're planted in? Will they become a competitor for nutrients as they take away nitrogen and then cycle it? Most of our farms have actually seen a benefit. They're seeing increased nutrient availability as organic matter goes back into the soil. We've got some guys that are basically putting in anywhere from 10 to 30 ton per acre of organic matter every year. It's basically like having your own free compost. It's just a matter of getting it in and getting it to grow at the time that you need it to be there. The cost of these things is actually kind of a very broad topic. Some of our guys plant every other row, every other year. Some guys plant edge to edge on the tree rows every year. Some guys actually leave 3 to 4 feet off of the tree row to try and make the soil be a little bit warmer and radiate some more heat for the frost season. So costs are generally broad and kind of hard to define, but we've actually seen people using less fertility as the organic matter levels are coming up in the orchard, and they're actually able to get more out of the soil, have some healthier biology, and get the nutrients to turn over. As I talked about on some of the timings for things for frost, here's a walnut orchard with 2 different plantings in the same orchard. The picture on the left was the exact same cover crop planted on December 23rd. The picture on the right is the same cover crop that we have the SR mix planted October 10th. You can see the picture on the left is barely above your boots, not even to your knees. The picture on the right is all the way to the branches in the trees, probably above belt buckle, below chest height on those people. What was absolutely incredible, you'll see that the first bloom of the mustards and the first bloom of the daikon radish is visible. With 3 mustards in the mix, there's actually 2 more blooms to come. So if anybody was afraid of frost issues on this, this walnut orchard is Howard's, the variety Howard's, and so they're just starting to show signs of the catkins forming. We're probably 10 days to 2 weeks before we need to drive out there for any reason. But if this picture on the right was something you didn't like, you thought it was too tall, you wanted more room for your tractors, you could go through and mow it at 4 to 6 inches tall. And most of the items in here will continue to grow back because the heads haven't gone vertical or the plants haven't bloomed and set seed pods. It would be very interesting for some of our guys, and we've got some pictures that we'll show off that as we get done with our year-end trial here. This is one of our almond growers that planted a cover crop in October with solid set sprinklers and was able to water it and get it going. The picture on the left shows right in bloom time, the cover crop was just a little bit tall. It was right in the saddle of the trees, just where everything was starting to bloom. They were going to be doing some sprays. If you look on the picture on the left, you can see that the cover crop and the height on the tree, that was the last row right before they mowed it. They mowed the row to the right that same morning, and they came through and mowed every other row and then finished off mowing the other rows when they went back through the orchard. As you can see, the mustard was barely blooming, so it started to grow back, and the oats had not headed out yet, so they started to grow back. The bell beans kind of suffered a little bit. They don't like a haircut, but the peas did absolutely fine. They started coming back, and it got back to about the same height again where they mowed it actually yesterday. They mowed it before I got there to take the picture on the right, and they had the sprinklers on to get some water going and try and get some fertilizer pushed in. with everything going, these are two definite ways that guys have timed their different plantings and products. Some like to go earlier, some like to go later. It's a personal preference with everybody. I like to go earlier and try and get the bloom going as fast as possible, but everybody's got their reasons and everybody's got a recipe that works for them. This is a picture of one of my walnut orchards last year. This is also the same picture where the early and late planting from 2 slides ago was shown. Last year with our walnut blend, this would be the 3rd year we had it going in, we had almost 33 tons of green manure per acre that we mowed into the rows, let it decompose into the sprinkler system. When we got all done, you can see the orchard floor is fairly clean. We had absolutely no harvest issues with pickup. Everything seemed to go pretty good. The soil is incredibly soft. When you go through there was a couple rows over some main lines that we did not plant. That soil is definitely more compacted, definitely firmer. Our goal is to try and get that cover crop up every year. Our goal is probably to mow it in the next 10 days or so, maybe 2 weeks, depending on the weather. And so far, with the exception of we just haven't pruned yet because it's a little bit tall, we didn't want to run it over. Everything else is on schedule like what we would normally do. So with that, I would like to thank everybody for their time. I might be a little bit quick today, but I appreciate everybody listening to what I had to say. If anyone has any questions, I'd love to answer it. As we get through these trials, we're hoping to confirm our results through soil testing, biomass weights, and we're hoping to have a field day for this year before this year is over as our counties are starting to come out of their lockdowns. We're hoping that next week we'll be able to have an advance on that or some sort of news of some sort of update, and we'll get our results published as soon as we get everything finished up. We thank everybody for your time today, and if you have any questions, I'd love to answer them. Thank you very much.
Thank you, Brian. We do have a couple questions. The first question is, where did this trial take place?
I apologize. This trial was taking place in Glenn County, halfway between Willows and Hamilton City, kind of just west of Chico, on some fairly heavy clay soils, surrounded basically by walnuts and rice ground. So definitely a Northern California emphasis on these trials.
Okay. Do you do anything else to the cover crop besides mowing it to terminate?
So far, we have not. We've mowed it usually somewhere between 3 to 4 inches tall and then turned the sprinklers on for early irrigations, trying to maintain the saturation profile. The cover crop, when it's mowed that way, some of the items will come up the second time and we'll see some regrowth, especially where the tire tracks knocked it down and the mowers didn't catch it. But by the second pass, it's almost a clean floor as things start to dissolve. That's the beauty of the moisture content. We're usually running somewhere around 72 to 76% moisture in the samples when we mow it. It absolutely disintegrates quickly when we do that.
Okay. Can you tell us what seed drill was used for the trial?
We did 2 different drills. In our trial, we had a Great Plains that ran off a 3-point system that was 5 foot wide. And in the early and late pictures that you saw in the walnuts, the early pictures were planted with a John Deere conventional drill with the double disc openers. On the Great Plains that we used, it was a minimum till with the waffle cutter wheel in the front.
Okay, a couple more questions we got for you. Any trials done in drip systems?
We do not have any trials in drip at this time. We just got these going. I do have a trial that's in some drip that was planted. It's definitely behind this because it got planted right on December 23rd as well. But it is only about the top of your boots tall with no blooms yet. It's in heavy rice ground, and so it's still growing, but not enough to show differences in yet.
Okay. And if someone wanted to follow up to see how that trial turns out, we'll provide Brian's contact information.
We're hoping to get a trial where we can actually show these things off if we can get Glenn County out of the purple tier. Most of the counties around us has opened up. 2 years ago, we actually did a trial where we had a lunch and a field day, and we're hoping to build on that again. We're hoping that in the next week to 10 days, we'll be able to advertise that through OFAC and get everybody on board if anyone wants to see it.
Okay, and last question, I believe. Can forage crop be planted as a cover plan?
We actually do have some people that plant forage grains, whether it be oats, beardless wheat, beardless barley between the rows. They actually do quite well through the winter rains. Their root system is excellent for this, structure, 4 to 6 inch depths. Some people try and actually take it for hay cutting. They might cut it high, maybe 6 inches tall, to try and leave some root structure for water penetration and to keep that soil loose, and then still be able to take a cutting of hay off the top. I'm a big fan of the mulch and getting the biomass worked into the ground, but everybody's got their different processes. The only problem I have with forages is if you do try and hay, you have to be really careful with any hay equipment driving over main lines or soft ground and newly planted orchards. But we do have quite a few people do wheats, triticales, barleys. They stay fairly low through the early part of the year in frost season and then kind of continue to elongate now this time of the year for the next 2 weeks. It's been very popular in some areas.
Okay. Thank you again, Brian. We appreciate having you here today to talk about this project. Again, this project was funded by CDFA Healthy Soils Grant Project. And so if you'd like any further information about it, feel free, you can reach out to Brian. We've put his contact information in the chat panel on the Zoom interface. But once again, thank you, Brian. We appreciate having you here and helping us with this Healthy Soils project.
Thank you, everyone, for a great day. Hope you get some rain today. Good luck.
Yeah. So up next, we just launched a quick poll. If everyone could interact with that, that would be very helpful. I think we provide these polls back to CDFA, which helps us with the grant. So up next, we have Dr. Wyant. He's with Helia Ag Science, and he is currently the Vice President of Ag Science. So Dr. Wyant sits on our Western Region Certified Crop Advisors Board. He is also the Vice Chair of that board. And he has a talk today for everyone about soil health opportunities in organic production systems. So I'm gonna end that poll and let Carl take it away.
Thank you, Miranda. And thank you, Sarah, for inviting me to speak at the OFAC webinar. I greatly appreciate it. And thanks to Brian for sharing his information on the cover crops. That stuff's pretty cool. I think every time I see a talk, we've advanced that knowledge on using cover crops, to the next level. So super cool stuff. I'm excited to see it evolve. And today I'm here to talk to you about soil health and organic production systems. And it's not super data heavy. It's more conceptual. And I just kind of want to put a bug in your ear about why organic growers and organic production systems need to pay attention to the soil health piece. So I'll jump through my talk here, and I'm Miranda. At the end, are you going to share my contact information so folks can get a hold of me?
Yes, definitely.
Awesome, thank you. Excellent. So just a little bit about me. Here's my official headshot. It's actually from when I got married, so it's a number of years ago, and I looked a lot better. But that's what I use. But just to talk about my experience, I'm a soil scientist by training. I worked all over Eastern Colorado doing rangeland soil research and mostly just dodging rattlesnakes and cactus, which is a lot of fun. I've worked on Arctic permafrost, and then I moved to ASU in 2008, and I finished up my PhD in desert soil science. So when I finished up my PhD, my wife told me I needed to get a job. So I did, and I became an agronomist with a large ag retailer in the desert. And I covered from Phoenix to San Diego and got to experience a lot of organic crop production on the produce side with your cauliflower, your broccoli, and your romaines, and all the way to sweet corn and even small grain production. So lots of experience with organic production. And then I've been the VP of Ag Science at Helia since this past summer, July 2020. And prior to that, I was the Director of Ag Science in November 2019. So just a bit about me. And I just wanted to touch real pace. We're involved in the organic space. We have an organic microalgae product. I won't plug it here. I just wanted to let you know that's our connection into the organic industry. And I'll have an update with more details in April at the next webinar. So one thing I wanted to talk about is what is soil health? The soil health piece is I think kind of a buzzword: soil health, soil quality, whatever you want to call it. I'm going to use the definition that's kind of provides some nuance here to what soil health is, and that's this notion that the soil health is the interaction between organisms and their environment and the properties provided by such interactions. So healthy soils have a strong biological component in addition to the chemical and physical nature of that soil. So if you can imagine, healthy soils can promote better crop growth. And so if you walk away from this message in this webinar with kind of a key summary, when you think about soil health in an organic production system, think about the biological integrity of your field. How alive is that soil? How, to borrow a popular book on the subject, how teeming with microbes is your organic ground? And I think that's a good way to kind of root yourself in this whole soil health definition and make it a little more relatable to what you're doing. So if you have strong biological integrity of your soil, and really it's about the microbes, you can help focus on supporting plant growth. So just the key message there. I do want to show you an example. Here's an example of, what does soil health look like? This is farm soil. We ask these microbes to grow, and our grower standard is on the plate that's on the left. And you can see not a whole lot of biology there, not a whole lot of life growing in these inoculation marks. You got one colony. And for those of you who have your computer screens in front of you, I won't judge you if I can see you in the gallery. Squint real hard to see the rest of those colonies. But again, not a lot of biology. Again, a poor soil health score. We made a management change to this system. If you just move your eyes to the right, we made a management change to the system, and now we have a quantifiable difference in soil health, a much stronger biological integrity. So we've made management decisions and moved them into this realm where we have a higher sort of microbial score or microbial population and diversity. And that's what I want to talk to you about, is how to identify those management decisions and then how to measure it so you know that you're on the right track for improving soil health. And I think this is an issue because if you look at most soil reports, they focus on chemical information, and that's all your ppms that are coming across, your phosphate levels, your potassium levels. We've all seen these before. A lot of chemical data, and it also focuses on physical data, things like this pH, maybe the CEC, how much nutrients that soil can hold, that sort of thing. I think the only real soil health measurement that comes on a typical lab report is very, very slight. It's this organic matter, this percentage point. That might be your only indication of, am I improving my soil health? Am I improving the biological integrity of the field? And that's where additional tests can come into play. So I'll bring that in at the end. I'm going to start with low-tech tests and then tests that are a little more expensive. So we can, so you have somewhere to get started. So yeah, so soil health data is missing. It's just not there. We're just sort of dipping our toe in the water on this soil health information, getting comfortable with the concept, understanding why we need to have improved soil health. And here, now the data streams are catching up. And more importantly, the interpretation of the data is starting to catch up to help us guide our management decisions. So here I'm gonna make the case for optimizing soil biology in organic production. So that's what I'm gonna launch into, and then I'll start talking about the tests that you can do to say, hey, I'm on the right track. I am optimizing soil biology on my field. So here's my key message. In organic production, organic nutrient sources, so stuff we're trying to feed to the plant, are often tied to carbon sources. And just think of the piles on the side of the field at the start of the season, whether it's chicken pellets or crumbles or some of your plant compost sources. Those piles have lots of nutrients, I'm sure you've taken those tests before, and you can calculate the pounds of NPK in a ton on an as-is basis or on a dry weight, however you want to do it. But those nutrients are tied into carbon structures. So if you want to get access to those nutrients, something has to break apart those bonds. And I'll show you some examples here in a second. But what I want to reiterate is microbes, your soil biology, controls that release rate. They control the decomposition of your compost, of your chicken pellets, or whatever you're using. Could be grocery store waste products, could be fish preparations, whatever you're using for your nutrient source, it's tied to carbon, and microbes break that down, release the nutrients to the plant. So I'll just show you two examples. Here is the monomer of a protein, and it's called an amino acid, and this is the general structure of one. And here we want the nitrogen in that amino acid, but something's got to break it apart first. Something has to chop up all these different sticks. For folks that remember nightmares of organic chemistry, this is the ball and stick models. And you have to chop these bonds and cleave away that nitrogen from the carbon so that the plant can pull it in. If you look at phosphate, here's ATP. This is how biological systems pay for work. They cash in their ATP. Lots of phosphate here. And there's certainly ATP in your compost. If you want access to those nutrients, you have to break them apart. And that's where your microbes come into play. They're the ones with the cellular machinery to break these bonds and to release the nitrogen, release the phosphate, release the potassium, and make it available for your plant. There's been a lot of work done where they look at release rates of compost and manures. And if you're thinking about, I put it out in the spring, how much is available to my plant? This depends on your climate. You know, warmer systems can release those nutrients more quickly than colder climates. But just for some general statistics of that pile you put out or that liquid product you ran, maybe 5 to 20% of the nitrogen contained in that is immediately available for plant use. The rest of it tied up into these bonds. For phosphate, you can generally get 10 to 40% plant available. The rest of it is tied up. So if you can put these microbes to work and go with some management practices that help boost their population, boost their diversity, you can help speed up that release rate and better match your plant nutrient curves. So here's the thesis. Microbes control decomposition. And our goal as crop managers and as advisors, take carbon-bound nutrients and turn them into plant-available nutrients. How do we get there? It's the soil microbes. They're the ones that are controlling it. They're the ones that are doing the work for you. And if you can get them going, put them to work, you can make this carbon-bound nutrients transfer faster to plant-available nutrients. But there's some caveats here. You're working with a living system now. So those microbes are controlled by temperature, moisture, and then the substrate properties. So what you're feeding them and what they have to feed on, that sort of thing. So lots of nuances here. But I think a great place to start is, how do I know if my management practices are increasing that soil microbiome? I have a goal. I want to turn carbon-bound nutrients into plant-available nutrients. But first, I need to boost the middleman, the soil microbiome. How do I know I'm getting there? So I'll talk about that here in a second. One piece to think about when you're thinking about microbial decomposition is that there's a considerable lag between what feels like ideal conditions to us versus how fast that soil is warming. So here's a climate graph for Atwater, California. I have some sensors in the ground, some temperature sensors in the soil, so I could calculate the difference in dates between the how good it feels above ground, what we would feel versus how warm the soils are. Most microbes really get going when you start hitting those 70-degree temperatures in the soil, and they have that narrow window they work through, between 65 and about 80 degrees. They love it. That's when they're prime. On either end of those spectrums, the soil gets too cold or too hot and dry, and they start to shut down. But one thing to notice is that on this graph here, we're hitting these beautiful temperatures, these 70-degree days in Atwater, starting in April. But if you look at your soil temperatures, we're not hitting 70 degrees until middle of May and into late May. So there's a lag there. So release rates are driven by those microbes. Those microbes are controlled largely by temperature. And I'm assuming you're providing moisture from the irrigation system. But your plant's trying to pull nutrients, but there's a lag. So if we can boost those microbial populations, get them going earlier, give them the right sort of set of food sources to eat from, we can make sure that this lag is not quite as pronounced. So just to kind of show you a difference, here's where we're at. It feels pretty good outside, this mid-March, late March timeframe. But if you track this gray bar here, that's the soil temperature that I have sensors on. We're not hitting prime conditions for microbial activity until a bit later, about a month lag behind what we're experiencing. So key messages: organic production systems are dependent on complex carbon-bound nutrients, and that's all the way from the pile, the pile that sits at the side of the field before it's spread, chicken pellets, compost, manures, just to reflect the diversity of the growers on the call. It even is reflected in the products that we use, the in-season liquids, fish preparations and the fermented waste, the soys, the corn steep liquors. Complex carbon-bound nutrients are all over organic production, and they need a partner to help break those carbon bonds to release the nutrients. This is very different. If you go to conventional agriculture, a lot of those fertilizers are salts. They have a positive ion and a negative ion. As soon as they hit water, they separate, plant can use it. It's a very much quicker release relative to this carbon piece that organic production has to deal with. So I think it's worth thinking about. And then finally, release rates of this decomposition driven by the microbes can lag, especially early on when those soil temperatures haven't quite hit those optimums. And so boosting your soil microbiome, increasing the middleman. Here's maybe the first time someone will argue for increasing the middleman in a supply chain. Increase the middleman by making some management decisions in that soil microbiome. But how do we do that? How do we improve our soil microbiology? Luckily, we have a number of great tools to do this. One key is to feed the soil biology, and there's a whole bunch of products on the market, that can do this. This soil microbial food provides that food source for the microbes, gives them something to eat so you can start building those populations over the season. Cover cropping, just to go back to Brian's talk, great way to boost your microbiome because those living roots, and you're maintaining living roots over the year, can feed the microbiome with the carbon that they release. Reducing tillage is another way. If we reduce tillage, we reduce the disturbance into those microbial communities, and we can keep those populations high. Mulches and compost, they provide a bulk carbon source and a nutrient source to the soil at the same time. But it's worth knowing that the carbon in the mulch and compost is often tied up in, like I've been talking about, in those long chains. So sometimes if you amend the soil with the soil microbial food and bring it in with your compost, great way to get everything going. And then finally, I read a paper a while back about irrigation management and microbial health. If you're drowning the microbes because you're over irrigating or you had bad distribution uniformity, or you're under irrigating and you're drying the soil out, that's going to directly impact your microbial communities. So just to tie into irrigation management, how critical it is for our systems. And here's a way to think about it in terms of keeping your microbes alive. So now that we've established this thesis of, okay, I need my partners in the field, my fungi, my bacteria, and I made some decisions to boost their populations, whether it's a food source, whether it's cover cropping, or it's more mulches and compost in the system, or you did some distribution uniformity fixes on your field. How do we know we're boosting the middleman? How do we know we're increasing the population of the microbes? That's a great question. So I'm going to go through some low tech options and then work my way into some actual laboratory measurements. So you can have a place to get started and to talk to your crop advisor about. So simple and cheap tests. All you need is a shovel and your senses to do a couple of these. When we're going to look at residue on the field, the soil surface, we're going to take that shovel and look at the soil profile. We're going to look at plant roots. And then really all the technology that you're deploying here are your senses. Not all of them. I'll caveat that. Mostly sight, smell. I promise you, the smell test it works, and I'll show you why. And touch. I haven't figured out a way to use taste and hearing yet in these samples. If any of you figure that out, maybe you've got the new guy on the ranch. Maybe have him do the taste test as a fun exercise. I don't know. I don't know of anyone who's done that successfully yet. So no taste and hearing, just sight, smell, and touch. A great way to get in these kind of tests in a really low cost. So if we look at our residue characteristics, what we're looking for is residue that has signs of being processed. And that could be your cover crop residue, maybe residue from the crop prior. And that residue should look like it's had activity on it. So it should be shredded or physically processed. It should be looked like it's starting to blacken and decompose. If you're looking for evidence of biology, things like earthworms are a great way to look for under that residue. If you ever have pulled up some residue and seen these cobwebs, those aren't spiders, I promise. That's fungi in your system that's decomposing the material. That's signs that you have a good fungal system. And to boot, if you have a good fungal system, you generally have a good bacteria system as well, because they do slightly different things. So here's just some simple tests. Go out to the field and look at that residue, look at that soil surface, and see what you find. If you take your shovel, I promise that was all the technology you needed, and you dig, you should be able to find soil that has good structure, good granular structure that crumbles under your fingers, when you press it a little bit. This is a good sign because soil microbes control this aggregation along with plant roots. The soil bacteria, they secrete these glues that help stick your soil together. The fungi create these webs that help tie your soil particles together, and your result is good structure. So it's an indirect way to infer I have a good microbial community. Also, while you're digging, you might as well look for earthworms. There's one right there. That's a great sign that your soil is teeming with life, and that you're on the right track when you have earthworm populations building. I want to go back to that crumbling thing. The soil should crumble when you apply a little bit of pressure under your fingers. If you need to hit the soil with a hammer, to get the shovel in the ground, or let's just go even more low tech, hit it with the back of your truck hitch, which is, I know some of you have had to do before. That's a sign that you need some more biological integrity in your field, and that you might have a compaction issue. So just think of this picture, think of that worm. Soil should be nice and crumbly, and that's an indirect way to infer that there's life there. Now, here's going to be the most controversial part of my talk. Smell the soil. I don't want to tell you what I had to Google image search to find the picture of this guy on the right smelling an auger, but the picture exists, and here we go. So smell the soil. And this is going to be probably the weirdest idea you come away with, but if you use your sense of smell, you can infer some sort of information about the biology of that soil. If that soil, you take your core, you take your shovelful, if it has an earthy, sweet smell, that is a sign that you're on the right track, that there's living entities in the soil. You've improved the soil biology. That smell is from the geosmin released from a group called actinomycetes, and that you can infer from that excellent habitat, soil structure, and water infiltration. If you need a reference smell, go down to the Lowe's and just crack open a bag of potting soil. Tell them I said it's okay. Maybe don't do that. But that's the smell you're looking for, that potting soil smell. That tells you have a whole bunch of life in your soil. If it smells bad, if it smells like metal, that's telling you have a wet soil that's dominated by anaerobic bacteria. You have a drainage problem. and that's going to impact negatively your soil biology. So you want it to be potting soil smell, not metal smell. If it smells like rotten eggs, that's another sign that you have some water issues, some drainage issues. If it smells like urine, you know, maybe double check that you're sampled in the right spot. It wasn't where the dog went. But if it smells like that ammonia smell, that means you have another issue with the drainage as well. So there's a lot of these bad smells are connected into drainage, and good drainage, good microbial communities have an earthy sweet smell. Poor draining soils tend to have bad smells because you're forcing the soil to move through these metabolic reactions that are not as common. The piece I find most often is no smell, when a soil doesn't smell at all. And that's more common than you think. You get as close to the soil auger as you can, and you sniff as hard as possible. Don't sniff any dirt up. Been there, done that. But you smell, and there's no smell. There's no earthy smell. There's no sweet smell. There's no bad smell. There's no smell. There's an absence of scent. And that's telling you something, that there's not a whole lot of activity in the soil biology-wise. That's telling you that your management decisions aren't promoting life. They're not promoting the sweet smell, and that you need to do something. So this happens more often than you think. And this is a common question. I smelled my dirt. I didn't smell anything. Well, okay, let's make some decisions on boosting your soil health and biological integrity. I'll go into some lab tests here, and this is going to cost money. The other ones were free, unless maybe if you lose a bet and you make your friend smell the shovel, maybe that's the cost. But these ones will cost you a little bit of money. And I just want to talk about soil sampling for biology. It's different from soil sampling for nutrients and pH, and making lime recommendations and gypsum recommendations, just because you have a living component. And I think what it starts with is good record keeping. Take your soil samples, make the plan prior, sample the same spot that you can year over year, so you can keep a running record. And there's a whole bunch of apps and GPS points and whatnot. What we're looking for is a trend, the increase in soil health and soil biology over time. Before and after practices are a great way to get a baseline and see how your management changes are going. Observations can drive your sampling program. When you see a field do really, really well, it smells great, that's a great time to go take a soil sample and check for the biology. If the field's not doing so well, see what's going on below ground from a living component and see where you're at. So I just want to talk about microbial soil sample sensitivity and just when you should sample. If you're looking for a good time to sample microbes, they're often most active in the late spring into early summer. So this April, May period on the West Coast, and a little bit later into our more northern reaches. So if you just follow this graph on the left side of the screen, make sure you're timing your sampling activity to match microbial activity. And one piece I also wanted to talk about is where you're sampling. Here's the crop, and if you're a organic tree grower or a fruit tree grower, nut tree, just picture the tree as well. In dryland systems, you typically want to stick to that first 6 inches, and you have a little more latitude on your distance away from the plants where you can sample. In irrigated systems, what you need to do is collect your samples near that root zone at 6-inch depth, but don't stray too far away from that emitter. Because if you go out into the rows, you won't find a whole lot of biology relative to that community that's right next to the plant, right next to the emitter. And you only need to do 6 inches depth. That's where most of your biology is. If someone tells you to dig a 3-foot hole to find bacteria, they might be pulling your leg. So late spring, early summer, and just keep in mind your distance from the plant based on your system, irrigated or dryland, and then and your depth. Stick to the 6 inches, 8 inches, you'll be fine. So soil moisture is important. If your soil is super wet, you're gonna have a weird result. If your samples are super bone dry, you're gonna have a weird result. So when you're sampling, try to keep your soil moisture relatively, reasonable, where it's a good moisture level for the plant, that's a good moisture level for the microbes. So one piece that we've come into is lots of labs are coming on board with these soil samples, and particularly soil health samples, where they're telling us information. Here's how alive your soils are. But one of the issues we're finding is that the labs are providing data, they're providing the tests, but they're not often providing the interpretation of what it means. So here's a real life world example of a lab I sent a sample to see how alive it was. And I have a CO2 burst test at 1.6 ppm. Okay, that's a result. I have some, measurements of microbial biomass. Okay, that's another result I can use in the future. And available nitrate for the plant, coming through that microbial system like we talked about earlier. Okay, 3.4 pounds an acre. Okay, that's a result. If I just left it at that, I wouldn't know what to do next because I don't have the interpretation. Here's where that comes in. Make sure your lab is providing interpretation. This is telling me on this sample, typical low biology field, low nitrogen mineralization potential. So all that compost I just put on without having a lot of biology, I'm probably not going to mineralize a whole lot of it and make it available for the plant. So biological activity is low, and there's also low organic matter. So lots of room to improve on this management practice because I have the soil test and the interpretation. So maybe 5 years down the road, 2 years down the road, these soil samples come back and they say, great biology field. Nice job, Dr. Carl. Your management decisions really paid off. Your soil health and soil integrity and biological integrity has improved. We've changed that system. I'll wrap it up here. Just for next steps, organic productions have a lot of tremendous potential for soil health because of how dependent we are on the microbes decomposing those materials that we put out. And we can employ soil management and just small management changes, like I mentioned earlier, cover crops, soil microbial foods, mulches and composts, tillage changes. We can boost that soil biology. And then soil tests can help provide those guideposts on what to do initially and then keep you on the right track. There's a lot of resources, and Miranda's going to put up my information. If you're interested in the testing part, contact your favorite lab. There is a chance that they are already offering soil health tests, but if you sign up, make sure that lab is giving the interpretation. You don't just want a page of numbers. You want a page of action items. What do I do? What does it mean? What is this telling me? So contact your local lab first. You already have the relationship with them. Chances are they have these tests and they can help you out in the right direction. Don't forget about your cheap low-tech tests, smelling soil, digging up some dirt, taking a look at what's in there and looking at those residues. Something you can do today after the call, unless it's raining in your area. Don't do that. But there's also some real official stuff out there. NRCS is getting in on the soil health and quality. The Soil Health Institute and the Soil Health Partnership have great blogs and great information on how growers are integrating soil health management practices into their everyday programs. Excellent resource to check out. And then if you want to hear about Phycoterra and our soil health messaging, I also write a blog. And with that, I'll take any questions. I have a general email address on the screen there. And Miranda, I think just put my contact information in the chat box, and I'll take any questions, and I look forward to hearing from you. Thank you for your time.
Thank you, Carl. So we do have several questions for you, and we did put Carl's information in the chat box. So after the webinar's over, if you have further questions, you can reach out to him. For now, the questions that we do have, what are your, and I might butcher this, what are your thoughts on the Haney test? Yeah,
great test. Haney test has been around for a while, so there's definitely some interpretive value attached to it. The soil health metrics and testing arena is evolving quickly. So generally the tests are coming out without the backup data to support the interpretation. But the Haney test is a great way to look at soil. You get the CO2 respiration, how much CO2 is being breathed off the soil when you wet it. That's a great way to measure microbial activity indirectly. And then you also get some nitrogen tests from that and some other soil information. So it's kind of a nice one-stop shop to get a bunch of different information and help you make decisions. So if that's the test you like, I say keep using it and keep that in your back pocket, in your toolbox to help you out when you're making your management decisions.
Okay. Next question. For your soil health test example with poor results, what would you recommend applying to improve the soil health on that field?
Yeah, that's a great question. So I think where I would first start is observation. What might be driving my poor soil health results? Now I'm assuming that I've already corrected my soil sampling procedure. I thought about depth and I thought about distance away from the emitter and I thought about the seasonality already. So we'll just make sure that just for assumptions, we're all good on that. I think the next thing is observation. Is it this field poorly draining? Do my soil samples tell me there's low organic matter or low microbial counts? And that's a good place to start because there's a lot of roads you can go down. And I think I would start there and then figure out, does my field need more microbial food? Does it need more compost? Do I just need to fix the irrigation system? That might be as simple as it is. Do I need to go into cover crops? But start with that testing, start with that observation. And again, those low tech tests can help you just for not a whole lot of risk, go out in the field. I'm not going to judge you if I drive by the ranch and you're out smelling a shovel full of dirt. I promise you it works. It's weird, but it works. So that's where I would start. Thank you.
Okay. Next question is, are there benefits of using biochar? And if so, what role does it play?
Oh, great question. Biochars, if you think about a biochar, it's a big molecule. It's humic acids and biochars just size-wise are massive relative to some of these other things like amino acids and the ATPs that I showed earlier. So biochar, think about it as putting out an apartment complex in your soil. If you look at a biochar under a microscope, it's not a solid prill. It's got all these little holes in it and little rooms. And so when you put out a biochar, one of the advantages is you add more carbon to your soil, but you also provide habitat for your microbes to go hang out in and to start, living their life. So great tool. I like biochars. Yeah.
Okay. Next question. I'm gonna butcher this word because I do not have a degree in agronomy. Is mycorrhiza? What's the word? Do you know?
Mycorrhizae.
Okay. A part of the measures.
Yeah, you can. So let's just back up. Mycorrhizae is a relationship that plants go in with certain kinds of fungi, and they can go into this partnership. The plant pays for work with sugars, and the mycorrhizal fungus, pay back to the plant increased water access below ground and phosphate access. So great soil biology question. You can indeed test for mycorrhizae and take a look at your mycorrhizal applications. There's products out there that you can use to boost your mycorrhizae, and you can no doubt test for it in your management decisions. Great question. Miranda, congrats, you did a great job on that pronunciation. That was good. I knew what you were saying.
Well, I butchered it, but fortunately you're smart. So what do you think of DNA measurement?
Yeah, DNA measurements, probably from my body language and the tone of my voice can read, they're great information sets to have. They're great data to have. Where I think the DNA piece is difficult is the interpretation. It's like, so what? I know who's living in my soil, but what do I do? And I think that's what needs to catch up on those DNA tests is, here's your result. Here's how it compares to your grower standard or your check field or the sample from 2 years ago, whatever you were doing. And here's what it means. So I think if you're going to go down the route of taking those DNA tests, and I'm assuming we're talking about the field soil DNA, not 23andMe. That's a whole other conversation. make sure you're getting some interpretation from the company that's providing the results to you.
Okay. What are some examples of microbial food?
Yeah, it's a great example. I work for Helia, and we have our microalgae microbial food. There's also molasses. There's some of the fermented grocery store waste products that are out there. There's fish preparations. I think I said molasses already. I'll say it again because I don't remember. There's a whole lot of microbial foods. And one piece that we've learned in our research is that, you are what you eat. Those microbial foods have different nutrient qualities, and what you're feeding your microbes will influence your microbial community at the end. So you want abundance and you want diversity. And I'll have a more specific plug on that piece when we get back together here in April. But I can help walk you through those food sources if you contact me at the information in the chat.
Okay, thank you. Also, I just wanted to comment that someone pointed out that they appreciate that you mentioned the simple old-fashioned ways of testing soil. So thank you.
I've had so many interns look at me like I'm crazy when I'm saying, "Hey, smell this." They're like, "What are you doing?" It's super old school. Smell the soil. I promise you, it's like a sense we don't use enough in as soil scientists to think about living things in soil.
Okay, we have several more questions. So what do you feel about the addition of chitin?
Chitin.
Okay.
Chitin's a good product. Chitin being what makes up the fungal cell wall, and it's also what makes up the exoskeleton in bugs. To take everybody back to entomology 101, chitin can be a food source. It has some additional properties to it. I know some folks have experimented with chitin-based products, but I haven't seen a lot of soil health results connected to it directly. If you can prove me wrong, please email me. I'm super interested in this. But just to be frank, I haven't seen a whole lot of chitin-based soil health results yet. But if it's working for you, I say use it.
Okay. What lab or labs do you use to quantify microbes in the soil?
That's a great question. I would start, because I don't want to plug any labs or promote any labs, start with your local lab. More and more labs are getting in on these soil health tests and providing interpretation. So start with your local lab. If they can't help you out, ask them to make a recommendation. Chances are they have a relationship with another local lab that's doing it, or they already know about it. As the customer, they should help you out. So I say start in your own backyard, ask your local lab, and see where they're at on soil health tests.
Okay. Next question. Do you have a preference of which type of microbial test, DNA or respiration?
Yeah, so I mentioned the DNA earlier. DNA is information rich. Those tests come back with just pages of notes for you to chew through. So it's information rich, often interpretation poor. So make sure whatever DNA test you're looking at is giving you the interpretation. On the respiration side, it's an easy test to run, but the respiration piece can be difficult to replicate, month over month or year over year, because of how sensitive the microbes are to just very local changes in the soil. So one day you might strike it while it's hot and you get a lot of CO2 coming off, high ppms. And then the next month you go out and things have changed and it's a lot lower. It's a difficult test to make any sense out of unless you're doing a lot of samples. So I like both. One of my favorite soil health tests are these tests that sort of integrate the living component with the physical component. So things like active carbon, that's a measure of microbial food in the soil. It tends to be a little more stable relative to the respiration, the breathing piece by the microbes. And then there's also nitrogen protein, which is a way to think about how much nitrogen are my microbes releasing for the plants. So those are the two I like, active carbon and protein nitrogen.
Okay. What types of nitrogen products help feed microbes in the soil?
That's a great question. So the nitrogen needs to be attached to a carbon source of some sort to feed the microbes. If it's just straight nitrogen, you're not going to feed the microbes. There needs to be something in that blend or in the jug, like a molasses or a seaweed or a microalgae to feed the microbes. So just make sure if you're not just putting out straight nitrogen, make sure you're putting out nitrogen plus carbon source.
Okay, this is another personal comment. JW would like to know that you're not crazy. He also smells soil.
Hi, JW. Thanks for confirming that you're not crazy.
Yeah.
And for supporting my soil smelling thing.
So what are your thoughts and concerns about whole orchard incorporation?
Yeah, there's been some great research recently. It's showing that orchard recycling is a good way to put a bulk carbon source in the ground and take all those tree trimmings and the wood chips, put it below ground so it can start to decompose. I think my concern is that source of nutrients is going to be slowly available. Just think of how much work it takes below ground to decompose wood and release the nitrogen, phosphate, and potassium. So you've got to have a expectation of this is going to take 4 or 5 years to fully release, maybe even 10 years if you're up north in a cooler climate. And so I think great potential. Just make sure you're feeding those trees and the crops early on while you're waiting for the rest to catch up to decompose.
Okay. This one's a little loaded. Why are organic products so expensive?
I don't have an answer for that. Contact your favorite sales rep and ask them that question.
Okay. What's your opinion of amino acid, nitrogen, soy protein, hydrolysate?
Yeah, hydrolysate. Yeah, you nailed it, Miranda. Nice job. Yeah, great nutrient source. Plants can pull in amino acids. They can also let the microbes decompose the amino acids and release the nitrogen directly. Great nutrient source. I think it's a good way to supplement what you're doing in season and supplement your compost programs that you're putting out, because just price per pound of nitrogen in a soy product is more expensive than a pound of nitrogen in a compost. So I think you need both to make sure your crops are being fed the right amount of nitrogen.
Okay, I believe that's all of the questions. So if any of our attendees have more questions for Dr. Wyatt, we put his contact information in the chat box, so feel free to save that, copy and paste it into your notes or something. But thank you so much, Carl. It was a pleasure having you. I really appreciated some of your funny anecdotes this time. And some of our—
Are you going to go smell the soil, Miranda?
I'm not going to smell any soil at all. Nope. I think I've told you this before. I have no desire to become a CCA or go back to school for agronomy in any way.
Okay, fair enough.
So, but we appreciate having you here and supporting the OFAC program and talking about healthy soils.
Awesome. Thank you, everybody. I appreciate your time and your attention, and I look forward to interacting with you. Thank you.
So Carl will be back at our next seminar. We'll put those dates up at the end of the webinar, but he'll be talking about some of his company's products at our next one. So up next, we have Dana Yount, who's with UCCE in Glenn County, and she's going to talk about climate smart agriculture. Oh, she's with Climate Smart Agriculture and Community Education, and she's going to give an update on the Healthy Soils program and talk about a statewide case study. So Dana, if you're with us, feel free. Hi there.
Hi, yeah, give me just a second and I'll pull up my slides. I'm not going to go in as in-depth as the last two presenters. This will just be kind of a quick overview for everyone who might be interested in applying for the Healthy Soils program that's coming up hopefully soon. So let me pull
Yes, so as we talked about before, the trial that OFAC did with Lockwood Seed and Brian was supported by the Healthy Soils program. And so just talking about, Dana's just going to give an update on that program and where it is statewide, and how, if you want to get involved in the future, you can.
Yeah, thank you. No, the Lockwood Seed, that's a great example of a demonstration project, and hopefully we'll have a few more in our areas, and we'll be able to go out and do these events in person. That would be awesome. So without further ado, I'll just kind of give a quick update and answer any questions, hopefully, that are floating around out there. So I guess I'll just do a quick introduction too. So I work in the Glenn County office located in Orland. I'm the Climate Smart Agriculture Educator. So I kind of help do all of the CDFA grants. Right now, obviously the hot topic is the Healthy Soils grant. And we can help with anything from implementation, helping with your application, and then taking pictures and invoicing and doing everything after you're awarded too. So if you have any questions, please feel free to reach out. I'll explain a little bit more about that at the end, and kind of what our team does and how we can help. So everyone knows what the Healthy Soils program is. It's been kind of a buzzword. Hopefully we'll see the round soon. But just as a quick overview, there are demonstrations and incentives programs. So the Lockwood Seed is, like I said, a great example of a demonstration. If you don't want to conduct field days, the demonstration just has a little bit more of research input as well. But if you just like to apply for one of the practices, I listed a few of them below. There are a lot more practices get added every single year too. So those are some of the practices as of last year. Whole orchard recycling is on here, cover cropping, compost. All the popular ones right now are fundable. So if you'd like to apply for your ranch or have any questions about this, you can go on their website too, CDFA Healthy Soils website, and look at the past year's kind of manual for it and just read through it and see what it involves if you'd like. But this is just released, so an update in the corner. This is of January of 2021. They've put these Healthy Soils practices on over 56,000 acres. And this is just through the Healthy Soils grant. This doesn't include farmers that are already doing this on their own. But they've funded, it's probably been out for about 5 years, maybe more than that. But they've had over 600 projects, almost 650 actually, which is great. And a total of just over $41 million. So a lot of popularity for this program. It keeps growing every year. And there's already been a lot of talk about it this year. So I'm expecting a very close round, I guess is the best way to put it, for this year. So if this is of interest of you, start thinking about it now. So there is money in the budget as of now. There is nothing going on right now. I know in the past years, they've usually had rounds about this time, opening in December, January, and kind of closing in March or April. That is not going on this year. Please kind of be on the lookout for it potentially late this summer. It could be July or August. It also could be pushed back a little farther, more to like November, December. Again, nothing going on right now. Budget has not been finalized. We don't know really anything more than anyone else does. But when we do, I would say just sign up for the email list on their website. That's the quickest way to get information right from that office. And when they release for public comment, and they release the RGA that comes out, which is kind of the manual for the grant, those will come out beforehand. Obviously, public comment happens beforehand. If there's any concerns or questions, they'll get those answered. And that will start before the next grant cycle. So just keep an eye out on their website for that. Let's see, yeah, so a lot of interest this year. We're already getting phone calls of people thinking that it's going on right now. So just kind of keep that in mind. It will probably be pretty competitive, but on the other hand, the budget does keep increasing for these types of projects. So I hope that's the case this year as well. Okay, so yeah, so the other part of our job is we can help with invoicing and kind of everything. If you have been funded or know someone that's been funded that's maybe struggling to send in their invoices or get a hold of someone, our team, myself and our team, we can play kind of an integral role as the middleman in trying to help send those papers in, help get things signed, take pictures, do all of that kind of great stuff on the verification end. So please reach out for help or questions if you need one of us to come out and assist with anything, or the local RCDs. This bullet point list below is also some of the technical assistance providers. If one of our team, one of our climate smart agriculture educators is not near you, a lot of the local RCDs have a technical assistance provider in their office as well. CDFA also has a technical assistant grant. I believe the first year was this last year that they started it, but there might be another provider that is funded through CDFA, so maybe check for that. Point Blue also has some technical assistance providers, and then CAF, I'm not sure if they have providers, but they at least might be able to point you in the right direction to find someone that can help you. Okay, so our group, there's 10 of us statewide. I'm kind of the more northern one that based in Glenn County, but go up to Tehama. And we've got gals down in San Diego and Imperial County. So a good span of the state, spread out a little bit. And we're kind of focusing on the statewide case study project right now, trying to get it kind of wrapped up before the next round of CDFA grants come out, hopefully in July or August. So we will see. We're basically just doing a simple cost benefit analysis of cover crops. If we could do in-person interviews, so that obviously helps a lot rather than doing this over the phone. So we were able to actually go out to one of our growers yesterday and get a lot of great information about him, and just see his kind of opinions on, do you like it? Is it working for you? Do you have any challenges? Any change in erosion or irrigation? It's a pretty extensive study. We're not, I think we've got 4 or 5 people that we're interviewing right now. Hopefully we can get a few more. But that's kind of what's in the work. We're working with a grad student at Davis too, who is helping us run more of the economic end of this. But just overall kind of a good feeler to see what's going on, and how are you managing your cover crops, I guess, and what cover crops are you choosing? So one of the gals in our office, this isn't related, but I just thought it should be highlighted a little bit. She made a cover crop selection tool, and this is from Shulamit Schroeder. She's down in Kern County, actually located in Bakersfield. There's a link for it here. But if you'd like to find this or just check it out a little bit more, you can find it on the UC Kern County website under her Climate Smart Ag bullet on the side. I think she's got it posted on the website there too. And you can download it on your computer. And I just wanted to show really quick, hopefully this comes up. Miranda or Sarah, are you guys able to see the Excel that I just pulled up?
No.
No, okay.
You probably have to
do a new share.
Yes.
Okay, let me try this. Is it
there? So we can see it, but there's no data in it.
Right. Yeah. This is just a blank one just to show examples. So she created this. I'm not really sure how, but this is an amazing tool for you if you just kind of like to play around with cover crops. Obviously her references, individual species. And once you fill out this first page, I just kind of left it blank because I wanted to show what it was, but you just enter your information in here. There's quite a bit that, and then you put in like what benefits that you'd like to see, kind of rank them from your main goals down to what you think you can maybe work on in a few years and maybe change your cover cropping. And it gives you kind of the names of plants below, shows you the calculations, and then goes into individual species. So it's a lot of information. It's a really great tool. So just as an option that is out there as well. But if you'd like to talk to her, she is a fanatic for cover crops. So I'm sure she would love to chat with you as well. So let me go back to my presentation. Okay, let's start it over. Okay, so the cover crop selection tool, yes, so that can be found on the Kern County website. And if you'd like more information, you can contact Shulamit, and I've got her information at the end as well. So just an overview of some of the things that you'll put in. So what are your main goals? This is kind of that section again. So let me go ahead and go over that. So our team, like I said, we're kind of from Glenn County down to Imperial and San Diego. We can help with questions. We do have some team members that speak Spanish, and we also have some translation services available. So if that is of concern of you, or if you know someone that would like that help, again, please reach out. We're here to help every step of the way. This is our team, and I can leave this up for a minute. If you find someone in your area that you'd like to reach out to, hopefully a few of these names look familiar. But this is kind of the team that's working on the case-wide project, and then we will all be here for the next round when the grants open back up. But if you need us sooner, please let us know. And I wrote down a few notes that I wanted to mention that I thought of after my presentation was done. I also just wanted to point out, if you, there's a lot of changes going on right now in the CDFA office. So you might be getting people that don't normally work in the CDFA office kind of reaching out to you about your grant if you've already been funded. So please be patient with that. They're, like I said, they just finished kind of rolling out with last year's grant agreements. I think the last got out in October, November. So there was no way that there was going to be another round ready by like January, February. It's just not going to happen. And with budget and everything, we'll push that off to, late summer or early fall. But if you know of someone that already has a grant agreement, particularly from the 2018-2019 round, and they're having some troubles, if COVID's affected anyone, just in general, there may be extensions available for your grants. I think they're doing a 6-month extension right now if you need it on a couple projects, and that's not just for healthy soils. We can reach out for SWEEP as well if COVID or something has affected you. So that's just as an option. Again, look for the public comment period, get on that email list if you'd like to stay notified about updates on the Healthy Soils program. And hopefully the RGA, which is kind of the manual for the grant, will be released soon too, so you can kind of start getting some ideas. But that's pretty much it. So I'll just leave it on that information if anyone wanted to get a phone number or something. But that is all I had. So thank you.
Thank you, Dana. There was a question: is there funding for almond orchard removal and whole orchard recycling?
Yes. So whole orchard recycling is the new practice that got funded last year. It's very new. There were a lot of questions about the process. There's some general information, and maybe I can pull it up really quick while we're talking too. In last year's RGA, that gave a few specifics on what exactly needed to be done with those chippings. So if you pull out your orchard, all of those chippings had to be incorporated back into that same plot of land. Nothing was able to leave off that premises. And there's some kind of instructions on incorporation depth. I think it has to go down to like 6 inches and a few things like that. Again, I'm assuming there will be a lot of comments about that in this upcoming public comment period, and that they'll kind of revise that RGA to answer some of those questions, because a lot of them we just got repeat questions. So I'm hoping that those will be included. But yeah, let me see if I can pull this up real quick just to kind of show where it is. Okay, this is for AMP. Okay, are you able to see the Healthy Soils page here?
Yes.
Okay, so this is just the CDFA Healthy Soils page. Hopefully it looks familiar to most people. Again, you can sign up for the emails right here if you'd like to receive upcoming news and information. If you want to see the projects that were funded for this last round, you can click this little blue link here. That was almost $22 million in projects right there, which is awesome. Let me move. Our little tab is in the way. So on the side here, maybe that says recipient resources, you can click that and it gives you all the information for the rounds in the past. So if you look under this 2020 and you see this request for grant applications, that's kind of what I've been talking about, the manual, the procedures. You can click on that and let me just type in orchard recycling. Again, here's a list of all of the practices based on grazing lands, orchards, any way that you want to break it down, they have it in here and what's allowable. And even if you want to stack these practices, you can. So for the one asking about whole orchard recycling, here's kind of the instructions or requirements, I guess, that you have to follow if you'd like to do whole orchard recycling. And this is on page 10. So hopefully that helps. This is all the information they have now. Like I said, I'm hoping, I'm assuming that there will be quite a few public comments about this process. So we should see more information soon.
Awesome. Thank you so much, Dana. We appreciate you talking with all of our attendees about this program.
Yeah.
If there's no other questions, we're right at our 10:30 time to end it. So thanks again, Dana. I'll let you go. Sarah's gonna pull up our slides. Just to reiterate, thank you so much to our sponsors. We can't hold these webinars and seminars without our sponsors. So this time we had New Era Farm Service, Tiger Soul, Organic Farms Fertilizers, and Brandt. Again, thank you to our speakers. We had Brian Loesch with Lockwood Seed, Dr. Wyant with Helia Agriculture, and then we had Dana Yount with the UCCE program. So our upcoming webinars, we're going to have one in April, and then we're going to attempt to do some in person in July, August, November, and then we'll round out the year with another online one in December. So if you have any questions or want to register for those, you can visit our website. It's organicfertilizerassociation.org /seminars. And so everyone that's here for your CCA hours, we're going to put up that QR code right now. And then I believe we will also email it to you, to everyone that attended after we get our attendee report downloaded. So if you're on the phone and you called into the seminar, we may not have your email, but you could reach out to us and we can get you that QR code. But it's on the screen now, so you should be able to scan it with your CCA app and get your credits. And using the QR code is the fastest way to get those into your CCA portal. If anyone has any other questions, feel free, you can reach out to our speakers directly. We put all of their information in the chat. You can also reach out to us through our website at the organicfertilizerassociation.org. And we're happy to try and answer any questions that you might have. If you have suggestions about future topics or things that pertaining to organic agriculture that you'd like to learn about, please shoot us a note and we're happy to try and find speakers about those. Other than that, we have a couple questions about the QR code never seems to work. So if you can't use if you're having issues with the QR code, after the seminar is over, we also submit an attendee list to the International CCA office. It takes them several months to get those attendees loaded into everyone's individual account. So it's a slower process, but that's another way that you'll get credit for the seminar. If there's no other questions, once again, thank you so much for attending. We know your time's valuable and we appreciate that you spent it with us. Have a great day, everybody.
Thank you.